Okay, everybody, I'm going to help you with this virtual gel electrophoresis lab, which can be found on page 7B of your handbook. Just in case you aren't able to run the simulation on your Chromebook, I wanted you to be able to see what we have here. So here is your start page where we are going to take this tube of clear liquid, and that liquid contains DNA strands of several different lengths, and we're going to figure out what those links are. So what you're looking at now are those different DNA strands of different lengths. And if they were as big as your shoelaces, you could sort them by hand into groups and measure them. But DNA is a molecule. It is not nearly as big as your shoelaces. You can't even see them under most microscopes. So is there a way to sort and measure the DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis. And now you should be able to answer question number 1. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. And this technique is also useful for separating other types of molecules like proteins. So now you should be able to answer question number 2. How does this work? What you're looking at is a gel. A gel is a filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it, and you can see that in the enlargement. In the previous animation, you saw them placing the DNA samples into holes at one end of the gel. These holes are also called wells. Electrophoresis is how the DNA is going to be pushed through the gel filter. You use an electrical current added to the filter that makes the DNA move. You should be able to answer question number 3. You can see from the animation that these short strands move through the holes in the gel more quickly than the long strands did. It all started up here. So these are the long strands. They didn't move very far, and these are the short strands, and they did. The short strands over time are going to move further away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. And in this way, the DNA strands in the sample sort themselves. You should be able to answer question number 4. Staining the sorted groups of DNA makes them visible to the naked eye. We can't see a single DNA strand, but we can see large groups of stained DNA strands. So these groups show up as bands in the gels, and those are the ones that are colored in this reddish pinkish tint. Those are called bands. So now we are going to run a gel together, and we are going to follow the steps which are shown right up here, starting with make the gel. So we need some powdered agarose, we need a buffer, we need a flask, we need a microwave, and we need the gel mold and the gel comb. This is kind of like making Jello, but you sure don't want to eat this kind of gel. We put a little bit of agarose here into the flask. This is a dried powder. Just like when you're making Jell-O, you start with a dried powder. Now we're going to add some liquid buffer to the flask. This is a salt water solution that lets electrical currents move through the gel. By the way, you should be able to answer questions 5 and 6 at this point. We loosely place some plastic over the top of the flask so that it doesn't boil over. We open up the microwave. We put the flask inside. Let's heat the mixture. Taking off the plastic wrap, we're going to pour this melted agarose into the gel mold. The tape holds it in. We're going to put the comb in one end of the gel. That's what makes the wells. Then we let the gel cool and solidify, just like when you put Jell-O in the refrigerator. This usually takes about half an hour for this kind of gel, but we're going to speed up the clock. And now we're going to take out the comb, and our gel is ready. So we're going to use the Gel Electrophoresis box to run our sample. We're going to use these materials shown here, starting with the buffer. Then we're going to take the gel mold, put it in the box. And now we need to load the DNA sample into the gel. So here's our list of things that we need. We have all of that stuff available. That's called a micropipette. We're going to suck up some loading buffer. We're going to add that to the DNA sample. It would be really hard to see them. Let me go back here. It would be really hard to see them if we tried to load it directly into the well. So the buffer has a dye that makes it easy to see. And it's goopy, so it makes the sample thicker so that it'll drop into the well instead of floating away. You should be able to answer question number 7. So now we're going to take this micropipette and we're going to put the DNA sample from the tube into the well of the gel. So let's suck up some DNA sample. And we're going to put that into the well. It's a little hole the comb made. This takes practice in real life. Now we're going to load up some DNA standard size. Put that into the well. This gives us samples of known length to use as a reference. That's question number 8, by the way. So now we're going to turn on the electricity. When we turn on the electricity, the black end gives us a negative charge, the red end gives us a positive charge. Together they will pass the current through the gel. DNA has a negative charge, so to move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. So let's plug them in. Click the location on the power supply that you want to plug in the cord. Let's turn it on. So now this is a look inside the gel box. We have tiny air bubbles coming out of the electrodes at both ends. This is proof that our current is running. So look at that, the DNA is moving through the gel, moving toward the positive charge. Remember, the short DNA goes faster. It's like the point guard on the basketball team. He can move faster than the big guys. Over time, the short DNA strands migrate farther from the starting point than the longer strands. We can't really see them, but we can see the dye. So now the gel is finished running. We're going to take it out, or it has been taken out of the electrophoresis box, and so now we got to analyze it. We need to stain it. The stain is going to bind to the DNA and show up under a fluorescent light. Here we go. I'm going to take it out of the mold, put it into the staining solution. Takes about an hour. We're going to speed it up. Let's put it on the gel on the UV box. Then turn that on. Look at there. Now you can determine the approximate lengths of the DNA strands in your sample. So we're going to compare the bands from the DNA sample with the bands of known length from the standard. This is going to help us answer question number 9. So right here, this is the standard. It says 6,000 base pairs here. So we should be able to determine what it is here. For some reason, it's not typing in. And then here, you could do the same thing. Look, here's 4,000, here's 3,000. So you should be able to determine what you think this one would be. Look, it's not all the way down here to 3,000. It's not all the way up here at 4,000. You should be able to determine what you think it is. So you're going to be writing that in. Wonder if it's about... And then look at this one. Here's 1,000. Here's 2,000. So wonder what this one would be. So I was close, but not completely right. So make sure you fill those in on question number 9. The last thing you need to do is the analysis section, the application section of the sheet, which involves answering questions 10, 11, and 12. And you should be able to do that on your own. Don't forget, you're going to upload that sheet.